Hello, everyone. My name is Ruth Reyes, and I'm a master's student in Physical Mathematical Sciences with a focus on nanosciences at the Universidad de Guadalajara. I will present my project titled Insulin Encapsulated in Chitosan Alginate Nanoparticles. We will cover the following sections: introduction, objectives, methods, results, conclusions, references, and acknowledgements. Diabetes mellitus is a chronic condition characterized by the body's inability to produce sufficient insulin, leading to abnormal glucose regulation. It's a significant health issue in Mexico and globally. Current treatments rely on subcutaneous insulin administration, which presents challenges like degradation and inconsistent efficacy. Nanotechnology offers a promising solution with nanoencapsulation potentially enhancing insulin stability, release, and bioavailability. This project aims to explore new diabetes treatment alternatives using chitosan and alginate due to their biocompatibility. Our objectives include encapsulating insulin to ionic cross-linking techniques, characterizing the nanoparticles using UV-Vis, Raman, and FDR-ATR, and measuring particle size via atomic force microscopy. The nanoparticles in this project are synthesized using ionic cross-linking. Three solutions are prepared. The first contains tripolyphosphate and insulin. The second contains alginate, and the third contains chitosan and acetic acid, all dissolved in water. The chitosan is placed under constant magnetic stirring at room temperature, while the TPP and alginate solutions are combined in a syringe. Using a syringe pump, and the solution containing the insulin is dripped into the chitosan solution. At this stage, we can perform DLS, AFM, and UV-Vis analysis. However, for Raman and FDR analysis, we need dry nanoparticles. To achieve this, we centrifuge or cool it, protect it with a cryoprotectant, sucrose, and then lyophilize it, preparing it for analysis and storage for future release studies. Regarding the results, let's discuss AFM. To analyze the samples, we deposit our nanoparticles through voice or via spin coating onto slate. Once analyzed, we obtain the following images. The images in column A correspond to nanoparticles without insulin and with concentrations defined as normal or base conditions. Column B shows chitosan and alginate reduced by half but loaded with insulin. As we can see, we are obtaining sizes ranging from 50 to 70 nanometers. Additionally, the sample in column A was deposited by dripping, while the sample in column B was deposited by spin coating, which may contribute to better particle distribution and analysis. Next, let's analyze the FTR. In this case, the nanoparticles were lyophilized for analysis, and we also analyzed the precursors separately. The insulin signal shown in blue was taken from Kien 2020. And if we analyze the spectrum of the nanoparticles, we see a peak corresponding to the CO double bond. But it's not very clear because it overlaps with the CO double bonds of alginate and chitosan. Therefore, we need other techniques to confirm that insulin is indeed encapsulated. We then turn to the Raman technique, where we also analyze the precursors. The image shows the signals corresponding to each one. In this case, the nanoparticles being organic do not present well-defined signals, but if we zoom in, we see three attenuated peaks. The peak at corresponds to the presence of TPP and the polymers present, which are alginate and chitosan. The peak at 1700 corresponds to the carbon oxygen double bonds, presenting insulin, alginate, and chitosan. At the peak at 3152 corresponds to the nitrogen, hydrogen, and oxygen hydrogen bonds of insulin, chitosan, and alginate. Regarding UVBs, this technique helps us determine the wavelength at which insulin appears, and we have this characteristic shape with peaks observed at 265 to 70 nanometers. We measured insulin diluted in the total amount of solvent used to form the nanoparticles, then measured the color to see how much was encapsulated during synthesis. And finally, at 24 and 48 hours, we found that if we let the system sit, more insulin is encapsulated, as observed by a decrease in absorbance. Finally, I would like to talk about the DLA's results. We have particles with a hydrodynamic radius of up to 1,000 nanometers, with other peaks to 100, 200, 400, and 800 nanometers. These large sizes detected by DLAs can tell us a lot about the system's stability, indicating the need of a technique to reduce nanoparticle aggregation, which is common for polymeric particles. One possible approach could be the use of ultrasonication or the addition of polyethylene glycol in the synthesis. In conclusion, despite being a developing field, this research shows that ionic cross-linking can successfully create nanoparticles suitable for insulin delivery, with potential benefits for diabetes treatment. We will continue with release studies under simulated physiological conditions. I would like to acknowledge my advisors, Dr. Celso Velázquez and Dr. Maria Luisa Ojeda, the Center for Nanoscience and Nanotechnology Research at Cuballes for their support with UV-based and FTIR measurements, the Renewable Energy Environmental Laboratory at Cuballes for their support with Raman, AFN, and DLS measurements, and the National Council of Humanities, Science, and Technology for my scholarship to continue my master's studies. Thank you all.